Hello guys, so it's a discussion for CBT2. Now the first question is, what is the approx number of base pairs which are associated with single nucleosome? The answer is going to be 146 base pair. So what is nucleosome? Nucleosome is actually the histone octamer which is wrapped by 1.75 ton of DNA. This is double-stranded DNA, and this double-stranded DNA wraps the histone octamer. This is histone octamer. This is negatively charged strand, and histone protein is positively charged, so there is a good electrostatic interaction, and it is 1.75 ton of double-stranded DNA which incorporates 146 base pair. It's a fixed number which you should know, and that is what is the question here. So 146 base pair is going to be there in the DNA portion which is wrapping around the histone octamer in the nucleosome, in single nucleosome. So answer is going to be A in this. The second question is asking you, from here, okay. So second question is asking you, which is not a heme protein. So when you talk about myoglobin, cytochrome C and catalase, they all are heme protein. Albumin is a simple protein. It's not having any heme component in it. So answer is going to be albumin here. When you're talking about heme protein, other example also I can give you. You enumerate all the proteins which are actually heme protein. There is a long list of protein which are heme protein. One is hemoglobin, another is myoglobin. Then you have catalase enzyme, then you have cytochromes. Then you have peroxidase enzyme, Then you have guanylyl cyclase enzyme. Then you have NOS enzyme, nitric oxide synthase enzyme. So this is an important list of enzymes which are actually heme protein, and if you see, albumin is not the heme protein, globulin is not the heme protein, they are actually the simple proteins. Now when we talk about the next question, heparin. Heparin is not a glycoprotein, not a lipoprotein, not a simple protein. Heparin is a glycosaminoglycan, which otherwise is called mucopolysaccharide. We have got 7 names in fact under this particular heading. One is heparin, another is heparan sulfate, then you have hyaluronic acid, then you have chondroitin sulfate and dermatan sulfate. And these are example of mucopolysaccharides or glycosaminoglycan. Dermatan sulfate, then you have keratan sulfate, type 1 and type 2. So these are 7 types of glycosaminoglycan. So heparin is a glycoside. It's not a protein, it's a glycosaminoglycan. Next question is asking you, true about NADPH except produces ATP in RBC? No ways. NADH cannot produce ATP in RBC. NADPH to dur ki baat. NADPH never produces ATP in any cell. NADPH is a coenzyme which is actually donor of reducing equivalent of for reductive biosynthetic reactions. This has got no role to play in ATP production at all in any cell. It is NADH which produces ATP. So this is absolutely wrong statement. So all of the following are true about NADPH except produces ATP. So this is the answer in this question. Helps in reductive biosynthesis, true statement. Formed by G6 glucose 6-phosphate dihydrate or G6PD, that is HMP shunt enzyme, rate limiting enzyme. That's also true statement. Help in maintaining the RBC membrane structure. That's also true by detoxifying the free radicals. Then NADPH protects the membrane. So answer for this question is going to be option A. Next question is talking to you about essential pentosuria, and you remember it's a though essential disorder, though it's a benign disorder, but for your questions it's actually a very important question. Essential pentosuria is actually associated with uronic acid pathway, where the enzyme xylitol dehydrogenase deficiency is seen to be associated with excretion of L-cellulose in the urine. When the L-cellulose starts coming out in the urine, it's going to be pentosuria. So this xylitol dehydrogenase enzyme deficiency in uronic acid pathway leads to pentosuria. Now welfare state, next question is welfare state, acetyl CoA is obtained from diet, which is obtained from diet, is least used for the synthesis of welfare state. So welfare state, we get the acetyl CoA from glucose. This is least used for synthesis of acetoacetate. Acetoacetate is a ketone body, and that's actually produced during starvation when the fatty acid oxidation produces a lot of acetyl CoA. Welfare state, you don't talk about ketone bodies. So acetyl CoA, which is produced from glucose in welfare state, is not responsible for acetoacetate production. Least used for ketone body production. Next question is 7th question, and in last couple of hours at the end of 48 hours of fasting, the main source of energy is ketone body. The muscle glycogen never supplies the glucose to the blood, so it's never a source of energy during starvation. Liver glycogen certainly supplies the glucose, but its stock lasts only till 24 hours, 18 to 24 hours, depending on the fed state of the individual, dietary habit of the individual. The liver glycogen is going to last only for 18 to 24 hours, but not beyond that. In this question, they are asking you the source of energy at the end of 48 hours. Nucleic acid does not provide you glucose or ATP. So acetoacetate, that is ketone body, is the best answer here. And you should know that when the starvation begins, the ketone body does not appear in the plasma or in the urine immediately. Rather, it takes time, and you should know that end of 48 hours you can have the ketone body coming out in the blood and then in the urine. Now coming to the next question, breakdown of one molecule of 16 carbon fully saturated palmitic acid by beta oxidation leads to formation of, answer is going to be 7 FADH2, 7 NADH and 8 acetyl CoA molecule. So it is 16 carbon, so it will be needing 7 cycle of beta oxidation. And in 7 cycle of beta oxidation, because each cycle produces you 1 FADH2 and 1 NADH, you'll get 7 FADH2 and 7 NADH. And when you're talking about acetyl CoA, the number of acetyl CoA moiety is going to be 8, because each cycle produces 1 acetyl CoA during beta oxidation, but last cycle produces you 2 acetyl CoA. So that count comes as 8 acetyl CoA in 7 cycles. So this is actually the list of energy gain which we get to have at the end of fatty acid complete beta oxidation of 16 carbon palmitic acid. Now coming to next question, and the question is asking you essential fatty acid. Alpha linolenic acid is the essential fatty acid in human. What is the reason? The reason is beyond delta 9 position, the human cell cannot introduce the double bond, and when we see, we have desaturase enzyme which can introduce a double bond at various positions in fatty acid chain. We have the desaturase enzyme which can introduce the double bond till delta 9 position, but not beyond that. So when we talk about linolenic acid, it's not linol, it's linolenic acid. It is 18 carbon having 3 double bond, and that 3 double bond is present at delta 9, delta 12 and delta 15 position. So this can be introduced, but these two cannot be introduced by desaturase enzyme, because we don't have desaturase enzyme which can introduce the double bond beyond delta 9. So that makes the alpha linolenic acid as a essential fatty acid. The another essential fatty acid is, you should know, is the linoleic acid. See the spelling difference. It's a linoleic acid. It's 18 carbon, 2 double bond, and that 2 double bond is present at delta 9 and delta 12 position. This also is essential for the same reason that it's having the double bond beyond delta 9, and that cannot be introduced by desaturase in human cell. Now coming to next question. Amino acid. Answer is going to be proline. Proline is the amino acid. It's characterized by presence of amino group in place of amino group, and it produces a particular structure in its molecule, and that is called pyrrolidine ring. So pyrrolidine ring, this name you remember, it's a characteristic finding seen in proline, and this is amino acid, not the amino acid, because it's not having NH3 plus, rather it's having the NH2 plus in it. And the special structure which is there in the proline is the pyrrolidine ring, which is made between the amino group and the side chain. And you should know one test is there, that's called ninhydrin test. You must have done in your first year MBBS, ninhydrin test. This test is actually done to differentiate the amino acid from amino acid. If the amino acid is having amino group, this ninhydrin will give purple shade, and if it is amino acid, it's going to be yellow in color. So this test will differentiate whether it is amino acid or amino acid, and amino acid they give yellow color in ninhydrin test. Now protein, next question is talking to you about purification of protein using ammonium sulfate. Ammonium sulfate is a highly soluble compound, water soluble compound, and is having tendency to precipitate the protein when it is used in higher quantity. So because it's very much soluble, and if you know, like you take the protein solution, this is a protein solution, and there is a layer of water around this protein, and that is like making this protein soluble. When you add the ammonium sulfate powder in this protein solution, the ammonium sulfate will try to dissolve itself. In that attempt, it will deprive the protein of its water shell. It will consume a lot of water shell to solubilize itself, so the water shell will be deprived around this protein, and this protein will undergo precipitation. So this is called salting out. So ammonium sulfate is basically used to precipitate the protein from a protein solution, and that phenomena is salting out. You are adding the ammonium sulfate and you are precipitating the protein. You are making the protein become very obvious in that solution because it's been precipitated. Why it is precipitated? Because ammonium sulfate removes the shell of hydration around the protein. There are two types of this particular test. One is called full saturation test, ammonium sulfate salting out test, full saturation test, and another is half saturation test. You always remember that when we talk about albumin, albumin is precipitated at full saturation. When we talk about globulin, globulin needs less amount of ammonium sulfate, so it's precipitated at half saturation. So this also may be a potential MCQ. Full saturation is like, it means like you need to have more amount of ammonium sulfate for precipitating the albumin, because it's a small in size and having large surface area in 1 mole, and globulin is large in size, having less number of molecule in 1 mole and less surface area. So half saturation, it is undergoing precipitation. Now coming to the next question. Question number 12. I'm sorry, it is, yeah, 1, 2 is missing in this. 21st amino acid, selenocysteine, is derived from. It's a very popular question, and students, they tend to make number of mistake here, but don't be under any wrong impression. Selenocysteine is not derived from cysteine. That is why they keep on frequently asking you. Selenocysteine is actually derived from serine. It's derived from serine. Serine is a hydroxyl group containing amino acid, and in the side chain this oxygen which is there is actually replaced by selenium at the time when the serine is being transported by its respective transfer RNA to the site of translation. So serine is modified to give you selenocysteine, and this modification takes place before the serine is incorporated in the polypeptide. So on the way itself the serine is modified and it's converted to selenocysteine, and it's a It's a UGA codon on mRNA, which directs the selenocysteine incorporation in elongating polypeptide. And this is a recent question in AIIMS, like now only this Sunday AIIMS exam is over, and they ask you a very popular question: selenocysteine is a post-translational modification or it's a co-translational modification? Please remember, selenocysteine, which is found in the polypeptide at times, is not there due to post-translational modification, not due to post-translation, rather it is a co-translational change. It's a co-translational insertion of selenocysteine, because selenocysteine is produced in the cytosol itself at the time when serine is being transported on its respective tRNA to the site of protein synthesis. So the question was asking you whether the selenocysteine is a post-translational modification or not. You have to say that it was not a post-translational modification. It is not a post-translational modification, rather it is a process. Selenocysteine occurrence in the polypeptide is because the selenocysteine is incorporated there at the time of translation itself, because the serine was modified much before. Now coming to nicotinamide, it's derived from tryptophan. It's a very easy question and repeated question. The tryptophan is a very important amino acid. It's actually essential amino acid, and it's having number of metabolic pathway, and the most important metabolic pathway is one where tryptophan is converted to nicotinamide in a multi-step process which needs vitamin B6 to be present. So nicotinamide is derived from tryptophan. Coming to the next question. Next question is asking you beta oxidation of the fatty acid. We have seen in the previous question, the beta oxidation needs the cofactor FAD and NAD. You need cycle of beta oxidation, you get to have one FADH2 and one NADH. So beta oxidation requires cofactor FAD and NAD. Now the next question is citrate, which is used in the fatty acid synthesis. It is used via which enzyme? If you remember, the acetyl-CoA, because it cannot be permeated across the mitochondrial membrane, it takes the help of PC enzyme citrate synthase to produce your citrate. But this citrate, once it comes to the cytosol, this is cytosol and citrate comes to cytosol, the citrate is acted upon for this citrate to give again the acetyl-CoA which it has trapped. You need to have a cytosolic enzyme called ATP citrate lyase. This ATP citrate lyase acts on the citrate to convert it to its precursor molecule oxaloacetate and acetyl-CoA in the cytosol, and then this acetyl-CoA, which is now made available in the cytosol, will be utilized for the fatty acid synthesis. So you may debate that even citrate synthase is playing role. That's true, citrate synthase is playing role in the mitochondria. But the best answer, if one answer is to be chosen, the best answer is going to be ATP citrate lyase, because that is the enzyme which is directly going to snatch the acetyl-CoA from the citrate which is needed for fatty acid synthesis. So this is a frequently asked question, and you should know the answer for this question is going to be ATP citrate lyase. If it is a PGI type of question, they may ask you what all enzymes are needed to, for acetyl-CoA to make, to avail the acetyl-CoA for the fatty acid synthesis. What all enzymes are needed? What all enzymes need to be cooperating to avail the acetyl-CoA for the fatty acid synthesis in the cytosol? That time you can say A and B both are the answer. But in this one single response type, you have to mark B as answer. So I feel this is the discussion of CBT2. Thank you very much.